one another, what can you deduce about their genetic makeup? But that's all theoretical. Did we ever look at the genetic material? We didn't. We kind of guessed that they have a copy of the dominant, a copy of the recessive. This appears to be dominant, this appears to be recessive, this appears to be codominant. But we didn't really talk about DNA, right? So today, in the second and last lecture until I come back in April, we're going to be talking a little bit about DNA. Guys, for the interest of time, let's keep the questions to the minimum. You noticed it and I noticed it. Most of the time the answer is in the next slide. So please just be patient and see what the next slide brings before you raise your hands. If your question is not answered at the very end, you can ask, or better yet, come to my office hours. You can come to my office hours next Tuesday, even though I'm not teaching next Tuesday, to come and see your exams, as well as to ask questions about today's lecture. Even though I won't be here with you, remember, study every day. Don't let the material pile up, please. Okay? All right. First of all, we said that something is being carried over from the parent to the children, right? And what is that something, knowing what you know about biology? DNA, right? So, how do we even know that the genes that are being transferred from the parent to the child is even in a chemical composition that we call DNA? How do we know that? What is the proof? This part of the class, the second lecture, is going to focus on many different seminal experiments that were carried out in early experiments in DNA work, in fact. So even if you are averse to seeing summary figures of experiments, stay with me. They're actually quite fun to discuss and to discover. Okay. So by the 1920s, oh God, that's already 100 years ago. We're all getting old. It was known that chromosomes consist of DNA and proteins. Can anyone name a protein that exists in chromosomes? Yes? Histones, but there are all kinds of other proteins that we didn't talk about. There are non-histone proteins too. But basically, there was this big technological advancement, if you can call it that, which was a dye, a stain. In fact, there are many different stains that would stain the DNA. Okay? And that was kind of circumstantial evidence that DNA could be the material that's being carried over. That's the genetic material. It was in the right place. It was in the nucleus, which was encouraging. It showed some variation from species to the next. It was present in the right amount, whatever that means. So they said maybe DNA is what is being transferred. That's what's that's what's carrying the information. So they carried out this experiment. I know it looks complicated. We're going to do this together, okay? So, what do you see in experiment number 1? You see a mouse, and it got an injection. And what happened to the mouse? It unfortunately appears to be dead. Why is it dead? Because what we injected in it was a virulent strain, okay? A virulent strain. So, a virulent strain of what? Of Streptococcus pneumoniae. So this is the thing that causes pneumonia. So there are two strains for it. One that's deadly, that's virulent, and the other one that's not. What are the differences? Why is one deadly and the other one isn't deadly? Well, S stands for smooth and R stands for rough. The smooth strain is deadly and the rough strain is not deadly. Why? Because the immune cells have a difficult time holding on to the smooth pathogen, so it escapes and continues killing the organism. Okay, it causes damage, it kills the organism because the immune system can't really tackle it. It's smooth, it's slippery. The rough one, the rough strain, doesn't have that smooth coat, so it's easily captured by the immune system, so it's not a big deal to have the R strain. If you're infected by the R strain, you're going to be okay. And that's what they did in experiment number 2. They took the R strain, they gave it to the mouse seems to be fine. And afterwards, In both cases, they still killed the mouse here too to take its blood. And they looked at the blood and they found that, oh, there are no bacterial cells. Why? What happened to the bacteria that we injected? The immune system took care of it. Okay? But in this case, the immune system couldn't take care of it. So you still see the S strain in the blood. Does that make sense? Good. Next, they did the following. They took the virulent strain, which is the S strain, it's the deadly one. But before they injected it to the animal, they heat inactivated it. So they boiled it, let's say. What happens when you boil something like that? You end up killing it. That's why, if you have something that's very dirty and you think might have bacteria, really put it in the antibacterial sanitary cycle on your washing machine and let it boil the hell out of that. And that's what they did. They really boiled that thing. They heated it up. So what happens to the S strain? It died. So can it still cause damage to the organism? No. So this mouse is lucky until they kill it and take its blood. And they found that there are no bacterial cells. Why? Because these bacteria were killed before being injected. So far, so good, right? It's kind of straightforward. Now, why is this even an experiment? It seems almost worse than my science fair project. What did they do next? And this is the cool part, guys.
They added it to the living cells.
Exactly. So what they did is they took the S cells that they had killed and they mixed it with the living cells. Now, what happens to bacteria? Bacteria can exchange genetic information without necessarily making babies. It's kind of cool. What can happen when you put two bacteria together? They can give each other genetic information. So in this case, what happened is that even though this S strain was heat inactivated, when you mix it with a live R strain and inject it in the animal ended up dying. They took the blood, and what did they find? S strain. It's but the S strain was dead. How did the S strain come back? When we injected just the heat inactivated S strain, we didn't find bacteria in the blood. How come we're finding it here? Something from the S strain, genetic material from the S strain, end up going to the R strain, which normally is not deadly. Does that make sense, guys? Okay, so this was very interesting. So they wanted to know what is the chemical nature of this thing that's going from the dead S strain to the R strain and transforming the R strain to an S strain, giving it that S strain property, which is the property to build that slippery coat. So what they did is the following. So they took the S strain, which is the virulent one, they heat inactivated it, and then what they did is they just removed everything else and they filtered everything. So they only have this solution here, okay? And then what they did here, they ended up treating it with different enzymes. In which case they gave, in the first case, they gave it an enzyme that kills what? RNA. In the second case, they gave it an enzyme that kills protein, and here they gave it an enzyme that kills DNA. And their question was, if we kill RNA, can we still do this transformation? Can we still make the strain deadly or not? Okay? So, when they destroyed the RNA and took that solution and added it to the R strain, which is not virulent normally, they found that things are, transformation is happening just like it happened in this picture. Okay? We have virulent S strains, and the mouse, will the mouse live or die here? Die. Okay? It's virulent. They did the same thing with proteins. So they got rid of proteins. So they got rid of the proteins before adding it to the R strain. And then they saw if it's virulent or not. What did they find? Is it virulent or not? Virulent. So getting rid of RNA doesn't make a difference. Getting rid of protein doesn't make a difference. Does getting rid of DNA make a difference? Indeed it does. Okay? And that was the experiment, that the transforming principle, the component, the chemical that is transforming the non-virulent R strain to a virulent S strain is actually DNA. Because activity is not lost if you destroy proteins or RNA, but if you destroy DNA, everything is ruined. There are also a couple of other experiments that kind of point in the same direction. And in this case, there are experiments that have to do with, bacteriophages. Okay? So, this is a picture of a bacteriophage. And has anyone heard of a bacteriophage? Does anyone know what a bacteriophage is? Yes?
It's a virus that kills bacteria.
Exactly. So how does it do that? It's kind of very gruesome. What does it do? What does the phage do?
It goes and like attaches itself onto the cell surface, and then it injects its own genetic material into it, and then it kind of takes over the cell's like mechanisms. And
Like
it
machinery, yeah?
Yeah, and it gets the cell to start reproducing.
Making more?
Itself, and then
Right.
eventually it'll burst open.
Making more viruses, and then you have a bunch of new viruses. And look at how gruesome that is, right? So it's quite gruesome. And the experiment here was that if we don't know what is the component that's going into the bacterium to cause this, let's find out. We don't know what it is. Could it be maybe proteins or could it be DNA? Okay, we don't know. We're going to find out. How are we going to find out? Well, we're going to use radioisotopes. We talked about radioisotopes, I think, the first time we met, the first lecture. And we said that radioisotopes are not the common, they're not the same atomic number that's abundantly found in nature. It's an isotope of that. In this case, it's a radioisotope of that. And what they did is they so we're not going to go into details of how they did it, but basically they labeled the DNA with a phosphorus radioisotope and the protein with a sulfur radioisotope. Okay? And the idea there is that if you have labeled it with a particular isotope, you can find it later on no matter where it goes. You can trace it. So what they did is, so this is the Hershey-Chase experiment. So the goal of it was to know whether it's DNA or protein that's the genetic material. So first they did the labeling of proteins and DNA with radioisotopes. And, let me explain this in greater detail. And then what they did is they took this bacteriophage that's labeled and they let it attach to the bacterium. What happens with when the phage attaches to the bacterium, like you mentioned? It's going to inject the genetic material inside. So what they wanted to find out is which part is getting injected into the bacterium. So how did they do that? They let it attach to the bacterium, and then they put it in a little blender to shake off anything that's not inside the bacterium, okay? If you blend it too much, the cells are going to rupture. You're not going to blend it too much. You're going to blend it a little bit just to shake off anything that's on the outside of the bacterium. All the genetic information that's been injected into the bacterium remains in the bacterium. Does that make sense? That's the experiment that they did. And afterwards, they wanted to see what they're going to find inside. With the idea that if it's DNA that's the genetic material, what do they want to find in the bacterium? P32 or S35? P32. However, if it's protein that's being injected and that's the genetic material, what do they expect to find in the bacterium? S35, and that's exactly the experiment here. And what they found, of course, as you already know, was DNA that was in the pellet. Guys, in this picture, I don't know why it's like this. It depends on the edition of the textbook. But as you know, Supernatant should be pointing at the green part and not the yellow part. I don't know why in this picture it's a typo. It's in one of the 2017 version of this book. Yes.
Do we have to know like those specifics, like if it was looking for the S35?
Yes, just because this is kind of a seminal experiment.
Okay.
And also it would, just because it makes sense that it was the sulfur with protein, because proteins have a lot of sulfur and DNA doesn't. So there's a reason behind why those were chosen. Okay. And you can do this also with eukaryotic cells. You don't have to only do this with bacteriophage experiments. But you can basically get a eukaryotic cell and transfect it, which is a transformation, just like we did with our S and R strains. You can do this also in eukaryotic cells, where you can introduce a certain gene that is going to give it a certain property. Maybe if I transfect it with a gene, it's going to become resistant to antibiotics, or it's going to need a certain type of nutrient, or it's going to be independent of a certain type of nutrient, etc., etc. So you can actually do this also in eukaryotic organisms. That's how transgenic animals also can be made. That's one way to make transgenic animals. Now, we take for granted the structure of DNA. We know that DNA looks like a double helix, but of course this wasn't very obvious. And in fact, it was done by the effort of many, many people. And the reason Rosalind Franklin is highlighted is because she got really the short end of the stick and not enough recognition for her work. But she did all the X-ray crystal, or most of the X-ray crystallography for this. So, and she was the one who gave that crucial piece of evidence that says that DNA has that form. Basically, the way we can determine the shape is that the position of the atoms in this structure can be inferred by how the X-ray gets diffracted. Okay, that's why it's called X-ray crystallography method, and that was Rosalind Franklin's contribution. And basically, what was the conclusion from all these studies is that we have the double helix, which you already know, with the sugar phosphate backbone and the bases sticking out. We're going to talk about that in detail. So there were also other clues that were there for this structure. So we know that the chemical composition of DNA is it a monomer or a polymer? It's a polymer. A polymer of what? Nucleotides. What are the components of a nucleotide? We did this many times. So you have a sugar, which is either ribose or deoxyribose. In the case of DNA, it's deoxyribose. You have the phosphate, and you have a nitrogenous base. What are the possibilities? ATGC, right? Okay, so, and we talked about purines versus pyrimidines. We said that one of them is the fused rings. Which one, purine or pyrimidine? Purine are the fused rings, good job, and pyrimidine is the single ring. So the pyrimidines are thymine and cytosine, and guanine and adenine are the purines, as you remember from many lectures ago. Okay. So one thing that was discovered by doing a lot of chemical analysis is that if you measure how much of the A we have, how much, if we just separate them, A will always equal to T. How much A we have, if we put A on one side and T on the other side, it's going to be equal. The same thing with the G and C. The abundance of G is the same as C. It's interesting, huh? Does that hint at anything? Why are they exactly equal? Yes?
Because like the helix has equal diameter.
Ah, yes, we're going to come back to that. Because they need to match, right? It's almost like saying we want everybody to find a mate, right? And everyone to find, like everyone is in a hetero relationship. So the number of boys has to equal the number of girls, right? Okay, so that's what we are seeing here. So this is the Chargaff experiments that are very, very famous. So this is in the 1950s. Basically, they found that the amount of A equals the amount of T, the amount of G equals the amount of C. So at the end of the day, the abundance of purines is equal to the abundance of pyrimidines. Does that make sense? If A equals to T and G equals to C, A plus G equals to T plus C. Purines equal to pyrimidines. So we have the same amount of purines as we have the amount of pyrimidines. Okay? And knowing what we know about the chemical structure of DNA, people started making 3D models of potential DNA structures. And of course, Crick and Watson, or Watson and Crick, were the ones who are usually credited for finding out the structure of DNA. Okay, so is DNA double-stranded or single-stranded? It's double-stranded. So we say that they're double-stranded, but the strands go in anti-parallel directions. That's the only way they can fit. So we said that as polymers, the growth happens from 5' to 3'. We're going to come back to that yet again. But if one strand is this way, the other strand is that way, and that's the way they're going to fit. So it's a double-stranded helix. The diameter is uniform, which is something that was mentioned. So if you look at the DNA double helix, do you see this diameter or do you see that? The first one, right? As is obvious in the pictures. What does that tell you about the pairing of purines with pyrimidines? Do you think purines pair up with pyrimidines or purines? Purines pair up with pyrimidines, and pyrimidines pair up with purines, because remember, guys, the purines are two rings. Imagine if we have two purines and then two pyrimidines and then two purines, two purines, two pyrimidines. Is it going to be uniform? No. How is it going to be uniform if we have one purine, one pyrimidine, or one pyrimidine? Okay? So it is known to be a right-handed double-stranded helix. So they always say that the easiest way to do it is point your thumb up and your fingers, it's going in this direction. Okay? So how are these two chains held together? We said that purines and pyrimidines are held together, right? That's why we have that uniform structure. Well, first we have hydrogen bonding between these bases. So this is known as complementary base pairing. So you have one purine and one pyrimidine. But we also have weak forces, van der Waals forces between adjacent bases that help them stack together. Now, this is the very classic view of what keeps the double-stranded helix in that particular shape. However, keep in mind that this is still early days, even though we always draw it a certain way and we say we know the forces, but there were papers published last year doing new calculations and saying, in fact, the majority of the binding is not because of hydrogen bonds, whatever. But you're responsible to know what the textbook tells you. Some of it is a little outdated. So for your purposes, please know that even though science is changing every day, hydrogen bonds, van der Waals interactions. So again, in order to maintain the uniform diameter, we have one purine with one pyrimidine and vice versa, and we don't have two purines together and two pyrimidines together. Okay. So A is pairing with T with two hydrogen bonds. That's an example of that. C and G are pairing together with three hydrogen bonds. And every base pair has one purine and one pyrimidine. And you can see this is the anti-parallel orientation that we were talking about. So if we have one that's going 5' to 3', the other one is going 3' to 5'. They're anti-parallel to one another. Okay? And because of that, because of the orientation of this anti-parallel helices and because of the complementary base pairing, you notice that you have a major groove and a minor groove formed because of these nitrogenous base pairs. Let's take a look and see what we mean by that. So in this case, we have a purine here and a pyrimidine there, and here we have another purine, another pyrimidine, and here we have the pyrimidine on one side, purine on the other side. And you can see that we have different number of hydrogen bonds. So with CG we have 3, with AT we have 2, and also these are different structures. G is different from A, and C is different from T, etc., etc. So the way they're organized is gonna have different areas exposed for potential interactions, okay? So in this figure, what we've done is that we've shaded the atoms that are available for hydrogen bonding with other molecules. So remember, guys, at the end of the day, the shape of a molecule depends largely on how the different atoms are interacting with other atoms. And that's why it's going to bend a certain way or have a certain conformation change. Okay, so remember, because of this, the backbones of the two DNA strands are closer to one side of the double helix than the other. That's why we have the minor and the major groups, because we have multiple possibilities. We have G here, C there, C here, G there, A here, T there, T. Okay? And in each case, you're going to have different atoms available for hydrogen bonding. Okay. Genetic material has important functions to carry out, and the structure of DNA is very well suited to carry out these functions. Number one, DNA stores genetic information. We have millions of nucleotides, and the sequence of the base pairs encodes a huge amount of information. Unfortunately, I'm not the one who's going to be teaching you about transcription and translation. Next week, last year I was doing that part as well, which was a lot of fun. But I'm not going to be here next week, of course. So please remember what I'm telling you now to link it to next week. So the structure of the DNA is ideal to store this type of information, huge amounts of information. The way DNA is also, the structure makes it susceptible to changes in information, mutations. Even if it's a small change in sequence, it can change information significantly. The structure of DNA makes it ideal to replicate very faithfully, and that's very important because we don't want to have mistakes when we're copying our DNA. Okay? The DNA is the most important information, so when cells are dividing, you really want the cell to have that original information. So very important to have this precise replication during cell division. And we do this by complementary base pairing. We're going to talk about that in great detail. And finally, all this information eventually is going to result in a certain phenotype. Okay? And this phenotype is the result of transcription and then translation. Again, things that Dr. Holtzman is going to tell you about next week. Okay. In Griffith's experiments, what happened when heat-killed S-strain pneumococci were injected into a mouse along with live R-strain pneumococci? So, the S is the virulent deadly one, but it's heat-killed. But they mixed it with the live R-strain. What happened? First of all, did the mouse die or not? The mouse died, so it's not nothing. What happened to the DNA? It went from S to the R. So which answer is the right answer? B. And of course they have C and D just to make sure that you know that genetic information is going through DNA and not through RNA and protein. Yes?
Why did people think that it was?
It was protein, for example. Yes, so in fact this was interesting. A lot of people, so first of all, they had no reason to believe it would be DNA. It could be one or the other. But the main reason people were averse to the idea that DNA could be carrying all that information is, how many base pairs do we have? ATGC. It's not that much. They're like, oh, how can we have this amount of information with just ATGC? At least with proteins, how many amino acids do we have? At least that's 20. This is 4. Okay. Later on, Arthur Kornberg showed that DNA itself has information for its own replication, which is, I think, the real magic of self-replicating organisms, that it has enough information to know how to replicate. So what he did is that in a test tube, he put a bunch of DNA, he put ATGC, And he put an enzyme called DNA polymerase and some salts and a buffer. You don't need to know the detail of that. But basically what happened is that new DNA was being synthesized. Basically what we're trying to say is that if you have the original information, the original piece of DNA, and we give it more building blocks, we give it more ATGCs that it can use, and we give it the right enzymes and the right pH conditions, it can replicate itself. So a very brief refresher from chapter 4. Remember, guys, differences between RNA and DNA is one is single-stranded, one is double-stranded. And also, we talked about uracil versus thymine. So in RNA, we don't have thymine. Instead of T, we have U, essentially. Something to keep in mind for today and next Tuesday as well. Okay. So now we've come a long way. We've said that, first of all, we know that DNA is the genetic material, right? That's how information is going from one strain to the other or from one generation to the other. But how does DNA replicate? We said it has enough information to replicate, but how does it do that? So there were three models that were proposed. Bless you. There were three models that were proposed. One was known as the semi-conservative model. The conservative model is the second one, and finally the dispersive model. So let's go over what each of them means. So in each case, you have an original double-stranded DNA. This is fine, right? The first column is straightforward. It's just the familiar double-stranded DNA that you know of. But how does it end up making two double-stranded DNAs? How is it copying? Well, the semi-conservative replication says that it opens up these double strands, and each one has its own copy. So the red is the newly synthesized copy, and the blue is the original. So basically what we're doing, we're adding a copy for each strand. That means that we have a piece of the original in each of the replicas. That's what the semi-conservative model says, how it works. The conservative model says maybe not. Maybe we just look at this DNA double helix and make a copy of it using new materials. That's the conservative model. And the third one is the dispersive replication. It says maybe we just cut these up and introduce new pieces everywhere. So we have like a chimera, I guess. So we have 4 strands that are all mixed new with the old. And this looks like it's a tricky thing. How can we find out which one is the right way that replication happens? So there are 3 possibilities. In one possibility, we have this double helix. We're opening it up, and each strand is having a copy of itself. So for the 2 copies, we have some of the old and some of the new. With the conservative model, 1 copy remains the same, remains original, and an entirely new copy is synthesized from new materials. And the dispersive is that it's a bit of everything for each strand. Okay? Does this make sense, guys, so far? Yeah? How did they discover which one is the right one? Because for me as a student, I saw this, and I'm like, who was the smart person who figured this out? This is a complicated problem. We're going to come. So, Messelson and Stahl carried out a very seminal experiment that did this and found that the answer is the first one, semi-conservative replication. How did they do that, guys? So they used something known as density labeling so that they can tell apart the old strand from the new strand. What does density labeling mean, do you think? Does anyone have an idea or any guesses? Density labeling?
Like what they did with that blender?
With the radioisotopes?
Yeah, that they did.
Similar to that? Yeah, you're labeling a component. Very good. Okay, is that what you were going to say as well? Yes. Okay. So basically, remember with the blender experiment, we labeled either protein or DNA, right, with S or P, radioactive S or P. In this case, we're labeling nitrogen. And is this nitrogen heavier or lighter than the common nitrogen that we know? Heavier, because the nitrogen that we all know and love is N14, right? This is N15. So it's a little heavier, okay? So all the original DNA has what? This heavy N15, okay, that we can find later on to see where it is. So stay with me. I thought this was very tricky when I was a student. when I became a professor, I looked at it again and I said, why was I confused? It's so simple, but I don't want you to be confused, so please stay with me. So in blue, what do you think we have, the heavy or the light nitrogen? Heavy. So you can see here maybe, I don't know if you can see, it's 15. It says 15. So the original DNA double helix is all heavy nitrogen, N15. Okay? And then what we're going to do is we're going to give it the Lego bits so that it can make a copy, right? So everything that it has in the environment is not heavy nitrogen. The heavy nitrogen is only in the original DNA double helix. Very important that you understand that. Okay? All the building blocks it's using to make the copy has heavy or light? Light. Okay? And that's denoted by the red color. Okay? So what they found is that if you give it 20 minutes and look at what it looks like, you find out that the first generation all has an intermediate N15, N14. Okay? Everything is intermediate. And then, if you give it some time for the second round of replication, you see that some are intermediate and some are light. What does this mean? Okay, do you like it when people draw on the blackboard or no? Yeah? Okay, I hope you can see. I'm going to try to make it quick. But basically, instead of doing red and blue because we only have white, I'm going to do either dashed lines for the original parent and a real line for the copy. Okay? So originally, we have a DNA double helix, so two strands, both containing the heavy nitrogen. Does that make sense? You can't see? No. Okay. Let's turn on the light. Okay. Can you see now? Yeah. Okay. So, this is the parent, okay? So the two strands are identical, and the two strands contain what? And 15, okay? Alright, but now they're gonna replicate, right? So let's do the three scenarios. Let's do the semi-conservative and the conservative and the dispersive. It's the three models. Okay, so let's imagine it's the conservative model. In the conservative model, how is it going to look in the first generation after 20 minutes? We're going to have the original remaining exactly the same, right, in the conservative model, and the new one only with light nitrogen, nitrogen 14. Does this make sense, guys? Okay? So with the conservative model, what we're saying is that the original DNA double helix remains the same. We're using it as an example to make a copy. And the copy is all made from new materials. If we were to do this, what would we find? We would find one heavy and one light band, right, in our experiments. Is that what we found in the slide there? Did we find one heavy and one light after 20 minutes? No. That means that the conservative model does not appear to be correct. Okay? Now, let's imagine that it's the dispersive model. Okay? In the dispersive model, what does it look like? In the dispersive model, we're going to have again 4 strands at the end, two double-stranded helixes. We're going to have one that's right? That's the dispersive, and the second one something similar. You get the point, right? Right? This is the dispersive model after 20 minutes. So what weight do each of these strands have? Is this heavy, light, or intermediate? And what about this one? Intermediate. What does our PowerPoint say? After 20 minutes, we're going to get intermediate. So far, the dispersive model seems to be correct. Do you understand? Yeah. We're getting 2 intermediate bits, so intermediate band. That makes sense. Okay, how did they know that the dispersive model is wrong? Now let's see what happens when this replicates. Okay, so that's why we have the 40-minute time point. What's going to happen when this replicates in the dispersive model? Yeah? We're going to have more longer lines, right? Less dashes, fewer dashes, because so we're going to have maybe so on and so forth. So what are we going to get basically? Are these light, heavy, or intermediate? Intermediate, a bit lighter than these guys, right? But still all intermediate. Is that what we're seeing there at 40 minutes? No, at 40 minutes we're seeing two things. Okay, now let's do the semi-conservative model. Are you still with me? Okay. So of course you can do it with the process of elimination, but that's not fun. Okay. So what does the semi-conservative model say? The semi-conservative model says that each one is gonna make its own copy. Okay? So we're gonna have two copies. Right? So in one case we're gonna have one strand that's the original and the other strand that's new. And here too, one that's the original and one that's new. Is this light or heavy or intermediate? Is this light or heavy or intermediate? So does this agree with our 20-minute observation? Wonderful. Let's see what's going to happen at the 40-minute time point. 40-minute time point. What's going to happen to the first line? How is its copy going to look like? Another line. What is the copy of this going to look like? One dash and one line. And the same here, right? Is this heavy or light? Light. Is this heavy or light or intermediate? Is this heavy, light, intermediate? Is this heavy, light, intermediate? So you're going to have intermediate and light. Does that agree with our observations? Do you understand this? Okay. Alrighty, so basically that's what's explained here with colors as opposed to the dash. If it didn't make sense to you, spend a couple of minutes doing it with colors or meeting up with a friend and doing it with dashes, colors, whatever works for you, but make sure you understand this. Okay, so basically they did what we did. They tried to see what the possibilities are, and they found that this pattern of intermediate and lightweight would only be found if the replication was semi-conservative. Okay, we already talked about that. If it was conservative, the first generation would have both high and low. If it was dispersive, then the second generation would not have intermediate and light. Okay. Now we know that it replicates in a semi-conservative manner, but how does that really occur? How does the replication itself occur? Now, we discussed this in lecture 2 when we're talking about polymerization. And we said that when we were talking about the DNA polymer, which is a polymer made up of nucleotides, polymerization happens from the 5' to the 3' end. Why? Because we're talking about the sugar molecule here. So in this case, it's a deoxyribose. So it's going from the 5' to 3'. That's how it's growing. That's how the chain is growing. So keep that in mind. Anytime you want to add to the chain of the polynucleotides, so the DNA chain, you're adding at the 3' end. And what is this reaction? The polymerization reaction, what kind of reaction is it? Condensation or dehydration, because what are you releasing here? Water. You're releasing water, okay? But very important to remember that elongation proceeds from 5' to 3'. So, in order for DNA replication to occur, you need to follow these two steps. First, you need to open up the two strands before we can copy them, right? And next, we need to fill in the blanks. Basically what we did here, we first opened them up, and then we filled in the blanks. Okay, these are the two steps we're going to do. How are we going to know how to fill the blanks? How are we going to know how to make a new strand? What are we going to use?
The complementary base pairs.
What do we mean by complementary base pairing, guys? Yes. Exactly. Remember we said the pyrimidine and the purine binding in a complementary manner? So having information of one strand is going to automatically tell you what the other strand is going to look like. It has to be complementary. Okay? Yes? Yeah, so we're going to talk about that at the very end. I don't want to go into it right now, but just because it's not super relevant, but we can discuss. Okay. So, remember guys, we have these two anti-parallel strands. So if one is going in the 5' to 3' direction, the other one is going from the 3' to 5' direction, but polymerization itself is always proceeding in which direction? 5' to 3', and this is going to present a problem with it later on. So the enzyme that does this, that puts these complementary base pairs, is known as DNA polymerase. We're going to talk about that in great detail. Now, does this circular thing remind you of something you've seen before? Perhaps last week or the week before? Ori, do you remember what Ori was? Exactly, very good. Ori versus terror, right? Where it ends. Okay. So, this is a very simple prokaryotic chromosome. We said it's circular. And the way replication happens is from the origin site, which is known as Ori, not exactly original. But basically, what needs to happen is that this pre-replication complex, which is a bunch of proteins, they're going to come here and they're going to signal the beginning of this replication. And just like I told you last time, it's going to go bidirectionally, okay? So it's going to open it up in both directions. And DNA replication is going to start from the Ori site, but go in both directions, okay? So when you're kind of unwinding this DNA, this is known as the replication fork. We're going to see it a bit later as well. With eukaryotic chromosomes, they're quite a bit more complex than the simple, small, circular prokaryotic chromosome. So if you were to only have one origin of replication, it would take you forever to replicate this whole thing. So in this case, what do you observe? Multiple origins of replication just to speed things up. And again, in the eukaryotic chromosome as well, you're going bidirectionally from that origin. Again, to make things go faster. Okay, how do we unwind the DNA? We said we need to separate the strands before we can replicate, right? So how do we do it? We do it with the help of enzymes. For the purposes of this course, we're going to focus on helicase, but there are other enzymes that are involved. So, what's going to happen when we are ready to replicate a strand? So, imagine if we already have helicase that opened up the double strand. So what do you see here? A single strand or a double strand? You see a single strand here. Okay, very good. So you have your single strand here. And you want to make a copy of that, right? With the complementary base pairing that you all know of. How does it start? Well, the first step is to unwind them. We said that we have to open them up to reach this single-stranded DNA. We did that, done. Next, we need to start somewhere, okay? And that somewhere is the RNA primer, okay? This is very important for people who do molecular biology work. So the RNA primer is where things are going to start copying, adding the complementary base pairs. How does this even happen? Well, there is an enzyme called primase that comes to the original template strand, binds on it, and says, okay, let's start here. Let's build the RNA primer, and that's what's going to get things started. Okay? So the RNA primer in this figure is intentionally a different color, so it's made in green. So primase comes, binds to the template is in blue, right? Primase is an enzyme that comes, binds to the template, and starts making the primer, which is here in green. And once that is done, once this green bit is completed, primase has done its job. So what does primase do? Goes home, right? Primase leaves, primase is released. And instead of primase, we have now a different enzyme here. It's in orange now. What is this enzyme? DNA polymerase. What is DNA polymerase? What does DNA polymerase do from the name? It adds these DNA polymers, the bits, right, to make the new strand. So in red, you see what the DNA polymerase has done, okay? So what was the part that made, what enzyme is responsible for the green RNA primer part? Primase, and the red part? Polymerase. Okay? So we're always starting with this short sequence known as a primer. This primer still has to be complementary to the DNA template. And the way this happens is that a primase comes and does that. And after primase has done its job and the primer has been synthesized, primase can leave, and instead DNA polymerase comes and starts adding nucleotides. Remember, we're always going from 5' to 3'. We're growing it. We're always adding the new bits to which end? 5' or 3'? 3'. Please remember this, very, very important.
So the DNA, when the rubber like the parent DNA unwinds, and then those two strands that unwind become.
Yes, yes, you will see that. So let's wait a little, we're going to see that. Okay, so one thing I want you to appreciate is that just like you saw in the enzymes chapter, the enzyme itself is quite large. It can really hug the DNA and the Lego bits of ATCG and put them together. And this is a picture of that DNA polymerase. So it's much larger than the substrate. Okay. So to go back to this. So this enzyme polymerase, also let's go back to the palm. I don't know why all the examples they give you, which I find confusing because it's the same example with the hand and the palm. So basically they said that imagine that the enzyme is shaped like an open right hand, and the palm is where the active sites are coming into contact, and the fingers are where you're sensing the different nucleotide bases and you know what to add to what. Okay? Now, let's take a step back. We're going to do this like in many, many ways, so please be patient. Your questions will be answered. So we're starting with the parent DNA. So this is the double helix. It's still double-stranded. What do we need to do first? We need to open it up. How do we open it up? Okay, very good. Helicase. Where is helicase? Which color is it? It's this dark red color, right? So this is helicase. It comes and it opens up. It starts unwinding this, okay? And in order for these strands not to stick back together, because they really like each other, they're complementary, there are little proteins called single-strand binding proteins that stick there to make sure that the strands don't come back to one another and the fork remains open. We really want the double strands to be separated from one another. Okay, now we separated the double strands, we have individual strands. What do we need to do next? We need to generate the complementary strand, right? How do we do that? We need a primer. Who generates the primer? Who synthesizes the primer? Primase. So primase is here in this like brownish color. And then when primase does its job, in this case here, who comes and takes over? Polymerase, and starts elongating this, always adding to the 3' end. Okay? Now, this is the replication fork where the unwinding is happening, and you are left with two single strands. Now remember, guys, and this is very important, so stay with me. These two single strands are going in parallel or anti-parallel direction? Anti-parallel. So let's say this strand is going from 3' to 5'. What's the other strand going? Parent strand. 5' to 3'. Why do we care? Well, let's find out. Let's start with the top strand, okay? It's called the leading strand. Let's find out why it's called the leading strand. There's a very good reason for that. Okay, so this is going from 3' to 5'. And in order for it to have a complementary strand, the complementary strand should be going from which direction to which direction? 5' to 3'. It has to be complementary. Is it easy to grow things 5' to 3'? So is it easy to add things to the 3' end? Yes, very easy. And this happens very smoothly. Once you have your primer in place, polymerase comes, and you just keep on adding the little Lego bits in a complementary fashion at the 3' end. Very easy to do. That's how polymerization of nucleotides happens in the first place. Very easy to do. That's how the enzyme works. Now what happens with the next strand? It's already not looking good. It's called the lagging strand. So not good news. Okay, the lagging strand, the original strand is 5' to 3'. That means the copy is 3' to 5'. Can we add things to the 5' end? Can DNA polymerase add things to the 5' end? No, it adds things to the 3' end. So what are we going to do? Well, instead of the simple thing that we did here where we started with the primer and continued, we're going to put the primer not at the end but somewhere here so that we can add 5' to 3'. And then we're going to put another primer and go this way and another primer and go that way. So you can see we're going to put multiple primers. Multiple times the primase has to come, synthesize a new primer just because polymerase, DNA polymerase, can only add things to the 3' end. Okay? That's why this is called the lagging fragment. And it's not done in one continuous sequence, right? In the leading strand, the newly synthesized copy is just a smooth one sequence thing. With the lagging strand, look at it. It's bits and pieces. We're going to have another bit here, another bit there. These are known as Okazaki fragments. Okay? So with the lagging strand, things are a little bit more different. So synthesis of the leading strand is straightforward. It's continuous. All you need to do is lay down your primer and continue building on that. Why? Because you're adding to the 3' end. Polymerase does that very easily. However, with the lagging strand, we can't really add things to the 5' end, so we have to put the primer a bit before, do this bit, and then put another primer here, do that bit, put another primer here, do that bit, and so on and so forth. And these are the Okazaki fragments. So you're going to do it bit by bit in the lagging fragment and continuously in the leading fragment. Very important to know this. Let's look at it one more time just to make sure everybody understands. So, the lagging strand goes in the 5' to 3' direction, which means that the complementary strand is 3' to 5'. But it's difficult to add things to the 5', so what we're going to do is we're going to put multiple primers. We're going to be putting one primer here and adding to the 3' end. Another primer there, another primer there. So we're actually adding multiple primers and multiple fragments. It's not continuous at all. Okay? And once we've done that, okay, if you still have questions, okay, ask very quickly.
So is one
Okazaki?
fragment one structure space of a 3' to 5'?
It's always going from 5' to 3'. Always. Look, guys. Always going from 5' to 3'. See? Just where we're putting the fragments. Remember, DNA polymerase doesn't add things to the 5' end. It only adds things to the 3' end. That's why we're putting the primer here and adding from there. Otherwise, we could just lay down the primer here and add it there.
Oh, that's why it requires...
Exactly.
Okay.
Yes.
So, if the polymerase creates the strand on the 3' strand, what would that fragment of the polymerase create involved?
Again, what is it? Okay, you can ask me later. Okay. Now what happens afterwards is there's a little enzyme called ligase and comes and stitches all these fragments together. Okay? At the end of the day, they have to be continuous, but they were not synthesized in a continuous way. Instead, they were synthesized as Okazaki fragments. So now we have another enzyme known as DNA ligase that's going to come and connect and glue these pieces together. Okay, so the job of the ligase is just to make ends meet, right? To connect the Okazaki fragments. So let's quickly review the enzymes that are involved in DNA replication. First thing we need to do when we see our DNA double helix is to unwind it. Which enzyme does that? Okay, next what do we need to keep them open? What do? We put bits of proteins there, exactly, to make sure they don't close up again. Excellent. Next, what do we do? How do we start replication? Primase needs to synthesize the primer. We just need one for the which strand? Leading strand. For the lagging strand, we need to do this multiple times. So that's primase. After primase lays down the primer, what does it do? Goes home. And which enzyme comes? Polymerase, DNA polymerase, and it comes and adds those pieces, always adding to the 3' end. That's why growth always goes from 5' to 3'. And some sometimes things can be made more efficiently by adding this clamp. So there's a chemical structure here. We call it the sliding DNA clamp. I find it very unscientific. But basically, think of it as a chemical structure that's going to make sure everything stays together, and DNA polymerase can really do its job much faster, and it's closer to what it needs to replicate, okay? Because at the end of the day, guys, all these are chemicals. Chemicals interact with other chemicals, sometimes don't interact, sometimes move a little bit. So this clamp really helps hold things together and makes replication go much, much faster. Okay, Okazaki fragments, we said, are added to RNA primers to replicate the lagging strand. It's not happening in a continuous way. When the primer is removed, there's no more DNA synthesis that can happen on the complementary lagging strand because there's no more 3' to extend. So we're going to talk a little bit about that. So, which one is the leading strand? Which one is the lagging strand? So 5' to 3' is the parent strand. So the new copy is leading or lagging? Lagging, and the opposite is also true. So when we're doing it, we put the primer here, right, and we copy it there. But when the primer is removed, there's nothing that can really take its place because nothing can be anchored to it, because nothing can be added to the 5' end. So that bit is kind of gonna just be cut off after replication. So each time you're replicating in the lagging strand, there's a little bit that you're losing, which means that your chromosomes are getting shorter and shorter after each round of replication, which could eventually lead to cell death. Okay, so let's talk about that again. So, at the end of chromosomes in eukaryotic cells, there are repetitive sequences known as telomeres. And the point of these telomeres is that they kind of are protective because they can, be cut off and not damage things too much. It's a repetitive sequence. And in rapidly dividing cells, that's very, very important. Imagine if a cell, just by its function, needs to divide quite a bit, and each time it's losing a bit of its information, that's obviously not good. So you want to have that little bit of a protection at the end. And also, in certain cell types, like in stem cells, there's a protective mechanism to make sure that doesn't happen. This is another enzyme that you might find interesting. It's called telomerase. And what telomerase does is that it contains an RNA sequence, and it just has its built-in template. So it just comes and lines up here, and now we can make a complementary sequence here. You see? And we can keep on extending. If we have this, we can put a primer here and extend there. Does that make sense? So it's almost like a portable little strand to help start the primer system.
So telomeres like added substances that allow for further DNA replication. So that way.
It's like a protective mechanism, but eventually telomeres are going to be cut off as well. That's why in certain cells, like stem cells, you have this additional mechanism of telomerase-initiated addition to the end. So you can come back with your telomerase, with your built-in RNA sequence, make the complementary sequence leave, and then make the sequence to the complementary sequence. Does that make sense? Okay, come see me after class. Okay. The last part, we want to talk a little bit about mistakes that can happen during replication. The example I always give here is if somebody gives you a very, very precious book that you need to copy. Okay? Obviously, you have to be very, very careful with that because if you copy the original wrong, afterwards when you're printing out multiple copies, you're going to have many, many mistakes, right? All the copies are going to have mistakes too. So that initial transcription of the book has to be very, very perfect. And that's how it is with DNA as well. Bless you, mistakes can always happen. And DNA can also be damaged even after replication due to environmental factors, etc. So it's very important that there are mechanisms to repair such damage. The three mechanisms we're going to mention, there are many others, are proofreading, mismatch repair, and excision repair. We're just going to go over them in a very general sense. The first is very easy. It's called DNA proofreading. And here, DNA polymerase, when it's synthesizing the copy strand, while synthesizing, it also proofreads. So this is akin to the person, the scribe who's copying the book. While he's copying, he's also double checking, okay? So there's that proofreading function. So if it makes a mistake, oh, this yellow base was not supposed to be there, I'm gonna remove it and put the red one back, okay? So it's just going over your answers one more time in the exam. This is the proofreading mechanism, and it's done by DNA polymerase. Another thing that can happen is mismatch repair. This is where you made the copy, and then afterwards, there are many ways to detect this, you come and see that there was a place where it was a mismatch. Maybe you had a purine matched up with another purine, and that's wrong. So that's going to be detected, and that's going to be fixed. So this is very important for newly replicated DNA. So proofreading is happening as we're replicating. The mismatch repair is happening right after we've replicated. We're going over it one more time. So it's still quite young. This new strand has just been made. And what we're doing is that there are many mechanisms. We're not going to go into the details, but basically we're trying to see which pair is wrong. It's like, oh, this yellow one is not supposed to be there. And what we do is we get rid of it and put a new piece in its place. Exactly. And we said DNA can be damaged even after replication. It's not that it's just a mistake during replication that needs to be corrected. Sometimes maybe you were in the sun for too long, or whatever the case may be, and you can have problems with your DNA that need to be repaired. And here you can have excision repair. So basically, even after replication is done, you can come and excise the bits, either just the single base excision or just the whole section can be excised, and the correct bit can be added. And as I said, there are many other ways that this happens, but the point of it is that there is a very tight control of what the replicated DNA looks like. It really needs to be perfect because it's going to be used to make a message and then protein, tons of protein. We cannot go wrong here, okay? Now, in the lab, I do this all the time, and maybe some of you will have experience working in labs and will do it too. You can make copies of DNA sequences using a machine called the PCR machine. PCR stands for polymerase chain reaction. Polymerase you already know about. What is the polymerase chain reaction? In the polymerase chain reaction, what you do is you take a double helix. What do you need to do before copying? Open it up, and then give it the building blocks and the polymerase enzyme, and you're going to make the copy. And then what are you going to do again? Open it up again and copy it again, and many, many times, so that you can amplify it. That's the whole point, okay? That's what the PCR does. So you're starting here, and you're opening it up, replicating it, opening it up, replicating it, opening it up, replicating it, and you can repeat it many times, and in a very short time you have many, many copies of the DNA that you can study. Because it's very difficult to study one double helix, right? That's not enough information. That's not enough material. But you can amplify that using the polymerase chain reaction. The person who made this recently passed away. He was actually a friend of my in-laws. My in-laws, like, no very fancy people. I did not come from such a family. It was a shock for me. It's like, yeah, I know that guy. I was like, good for you. But basically, that's how this machine works. In each cycle, it needs to take the double strand, open it up, copy it, right? And then open it up again, copy it, etc., etc. Initially, when this man was trying to make this machine, he was faced with a problem. How do we denature proteins and open up proteins, usually? Heat. Now, if you heat up an enzyme, does an enzyme like that? No. So the problem is that you want to unwind the DNA, but the best way to do it in a lab is to do it through heat. Helicase doesn't work apparently in a lab setting. So you have to, he tried everything. You have to do it through heat. You do it through heat, what happens to the enzyme, polymerase? It doesn't work. It's like, what am I going to do? And he came up with an ingenious idea. He said there are some animals that live in very hot environments, and their enzymes, including their DNA polymerase, can work at high temperatures. So why don't we get their DNA polymerase and get it to work for our PCR machine? And that's what he did. So he went to hot springs bacterium, Thermus aquaticus, took their DNA polymerase and used that in the PCR reaction. The PCR technique, what can it do? Can amplify only very small samples of DNA, amplify several random DNA sequences, requires synthetic primers to flank the regions of interest. Let me go back actually before explaining it. Okay, because we didn't talk about the primer. Before replication, you always need a primer. Who adds the primer here? Where is the primer coming from? Right, but it's not coming from the system itself, because this is not in a living organism. The scientist is adding the primer. Okay, very important to know. So maybe that will answer your question. So what do we need to do in a PCR reaction? We definitely need your lab-made synthetic primer. If you don't have the correct primer, you cannot do a PCR reaction. Most of our students who do PCR in our labs, validating the primer takes up the longest time. 30% of the bases in a sample of DNA are adenine. What percentage of cytosine is present in this DNA?
30.
So, we said that adenine, the amount of adenine is equal to the amount of, The amount of adenine is equal to the amount of what?
T.
A equals to T. How much A do we have? 30. And so how much T do we have? 30. So how much are we left with? 40. And that 40 is divided equally between G and C. So what's the percentage of C? Very good. Okay, guys. It was a pleasure. I won't see you until April. Study well, please. You can still come see me at my office hour next Tuesday.
I have a question.
Yes.
The
Crystallography?
something, yeah. In this bullet, when it said like a purified substance can be made to form the crystals,
Right.
like I don't get that.
So don't worry too much about it. If you want to understand in layman's terms, think about it in terms of salt crystals, for example, right? NaCl is going to organize itself to have a crystalline structure. That's true for many substances, including the DNA double helix. So once it organizes itself in a certain crystal structure, you can find out what that structure is if you put it in front of, if you pass X-ray.